Good morning or afternoon or whenever you guys are watching this, students. And I hope that y'all had a wonderful Thanksgiving break. I hope that you're ready to learn and had some rest time in. This week, we are focusing on cell transport. Cell transport is extremely important for a lot of things in your body, and it's going to be our main focus this week. So, you are going to be watching two videos for your asynchronous lesson, and this is the first one. These videos are going to help you fill out your notes in your class activities section of your OneNote notebook. You will have a cell transport notes kind of Venn diagram to fill in, and this video, as well as the next one, will help you fill out those notes that are worth 10 points and due on Thursday night by midnight. So first, we need to talk about the structure of the cell membrane before we can even talk about how things get in and out of your cells. So obviously, your body is made up of millions and trillions of cells, and each one of these cells has a cell membrane, okay? I want you to picture this kind of section as just a small, tiny section of a cell. Imagine it kind of going around in a circle and completing the whole cell itself. So number one is referring to the cell membrane in itself, okay? So you're going to notice that there's two layers, and we're going to talk about this in just a moment, but number one is referring to the cell membrane itself. Number two is referring to a protein. So there are things that are embedded in your cells that will allow processes to happen, and one of those things is a protein, which is number two. Number three is referring to a lipid bilayer. So you can see here that it's pointing to two layers. So you can see kind of a little ball here with some little stringy legs coming off of it. And then you have the same thing on this other side. So this is what we call a lipid bilayer. And it is made of lipids, which we learned about all the way at the beginning when we talked about macromolecules or biomolecules. And number three refers to the fact that there are two of these layers. Number four refers to a carbohydrate. These kind of stick up on the top of the cell membrane. And number five refers to a transport protein. So a transport protein has what we call a channel in the middle of it, and it will allow things to pass through this cell membrane. Finally, number six refers to a single phospholipid. Okay, so one of these little guys right here with the little ball and then the little legs, that is a single phospholipid. So you could call number three actually a lipid bilayer or a phospholipid bilayer. You'll need to be able to know how to label all of these things and tell me the functions as well. So let's talk about some facts surrounding the cell membrane. So the cell membrane has specific functions. It first regulates what enters and leaves the cell. Okay, so it's going to allow molecules, very small molecules, to come in, and it also expels waste. It's going to provide protection and support. Okay, it keeps everything inside of the cell, the mitochondria, the Golgi apparatus, the ribosomes, all of those little organelles. It is protection and support for those things. Again, it is made up of a lipid bilayer, so two layers of lipids, also containing some proteins, and it is a flexible structure. So you can see this picture over here to the right, and you can see that it's moving a little bit. So the cell membrane is actually flexible. It's always fluidly moving. Those lipids are kind of moving around. It's not a solid structure. The proteins that I showed you in the beginning, they form channels and pumps, and their purpose is to help move materials across the cell membrane. So you can see here, which is kind of another image of what we just looked at in the beginning, you have protein channels that allow things to go in and out, okay? And they are just embedded in random places on the cell membrane to allow for those molecules to come through. You can also see some carbohydrates here, and they're in blue. Okay, notice that if you look at the structure of these carbohydrates, they look like the structure that we talked about when we learned about biomolecules. Carbohydrates serve as identification markers to help cells identify one another. So they kind of talk to each other and communicate, right? Your body needs communication methods in order to know what cells should be there and what cells should not be there. So these carbohydrates are identification markers and communication methods. One of the main points of cell transport, or things coming in and out of the cell, is this word called homeostasis. Homeostasis is what we call a balance that organisms maintain through self-regulating adjustments. Okay, so that is the definition of homeostasis. Make sure to write that in your notes. Essentially, a cell is what we call an open system. So things are constantly coming into the cell and things are constantly leaving the cell. Okay, so matter and energy, those important things that your body needs, or a plant needs, or an animal needs, will come into your cell, and the waste will leave your cell. This is just a cute little thing from the Amoeba Sisters video, which I will link. This is going to serve as your lecture as well as the next video, but Amoeba Sisters always breaks things down, so I will give you guys a link to that video as well. Another term that you are going to need to know in order to understand cell transport is the word permeability. The cell membrane is what we call selectively permeable. Okay, now what does that mean? This is also termed semi-permeable. And what it means is that it's going to let some substances come through more readily than others. So it's picky, right? It lets some things in and not others. It's kind of like the bouncer. Okay, so if you think about somebody that's like kind of standing at the door, letting people in, saying yes to some people, saying no to other people, it's kind of like the bouncer of a club. Okay, and in one of your assignments this week, you were or you will be asked to give me kind of an analogy. So I want you to think about kind of something in your head that you can refer to as the cell membrane, letting some things in and not others. So things move in and out of the cell, but how? The first thing that you need to know is the concentration gradient. In the absence of other forces, materials tend to, this red underlined portion is huge, move from an area of what we call high concentration to an area of low concentration. What does that mean, Ms. Myers? I don't know what you're talking about. So essentially, we're going to look at a diagram here. If you were to label the area of high concentration, I want you to think for a second what that would be. Would it be the left side or the right side? If you said it would be the left side, you are correct. So the area that had a higher concentration of molecules, that's going to be the left side. That means that my right side over here has a lower concentration. There's fewer molecules over here. So if you were to draw an arrow on the drawing right here, showing the direction of movement for this solute, okay, so these little purple things are what we call solute. If things move from a high concentration to a low concentration, what way would we draw the arrow? Would we draw it pointing to the right, or would we draw it pointing to the left? If you said that we would draw the arrow pointing to the right, then you are correct. Okay, so here's my cell membrane. This is going to be kind of a thing that you will need to think about. Membranes are just going to be popping up, but things will move from a high concentration to a low concentration. I want you to always think about this. High to low. Start to memorize it now. We're going to look at some other examples here. So you see my solute or my thing. It could be any nutrient, a salt molecule, a sugar molecule, whatever it is. And here's our cell membrane. Okay. So we're going to do a few more examples. For letter A, there is a higher concentration of solute molecules on one side of the membrane. For letter B, the solute molecules will move from the side of higher concentration to the side of lower concentration. This movement is going to continue until the concentration is equal on both sides of the membrane. Okay, so here we have high concentration, low concentration, or actually no concentration, and things will move automatically from high to low. And this will just keep happening until it's equal on both sides. Letter C is an example of equilibrium. So after things move from high to low, equilibrium is reached. So you can see I have an equal number of molecules on both sides of the cell membrane. There will still be movement in both directions. So this is really important to think about. So molecules, once they get to where they need to go, they don't just stay there. They're going to keep moving back and forth. You can see the arrow goes back and forth in both directions so that the concentrations remain equal. So this is what we call just movement from a high concentration to a low concentration until equilibrium is met. There are two types of transport. We will have passive transport and active transport. By the end of this week, you'll know the difference between the two very clearly, I hope. In passive transport, there is no energy being used to move molecules across the membrane. You can pause this video and put this in your notes for passive transport. No energy. No energy being used. Energy is a big deal when it comes to transport. One type of passive transport is called diffusion. The definition of diffusion is the spreading out of molecules from an area of high concentration to an area of low concentration. Here we have an example. We've got a little bag in some water. Start getting used to seeing these examples. We're going to do some of these examples in class, pretending that we have a cell in a beaker. Okay, so this is my beaker. This is my cell. We're using kind of a bag as an analogy and some water. So I have a 20% salt solution inside of my bag, and I have a 5% salt solution in the water, which is outside of the bag. The first thing that we need to consider, so we're wondering in which direction will the salt molecules move? We need to consider the percentage that we're looking at. Okay, so we've got 20% and 5%. So if we are moving from a high concentration to a low concentration, we are going to be moving out of the bag. Okay, or my salt molecules will be moving out of the cell or the bag. Okay, always high to low. So if you're doing these in word problems and you're writing them down on a piece of paper, I highly recommend looking at those numbers, comparing them, maybe even writing them right next to each other, and always drawing an arrow from the bigger number to the smaller number. Okay. So here we went again from the high side to the low side. So out of the bag and into the water. And again, diffusion is the spreading of molecules. So in this example, salt molecules from high to low concentration. Another type of passive transport is osmosis. You might have heard this term before. Osmosis is the movement of water across a membrane from an area of high concentration to an area of low concentration. Now, the difference between diffusion and osmosis is simply the term water. Okay, so again, diffusion is just any old molecule. It could be sugar, it could be salt, it could be potassium, anything. Osmosis is strictly water only. Here we have another example with my bag in the water. And again, I have my 20% salt solution and my 5% salt solution, which is what we saw from the first example. The only difference here is that I know my salt concentrations in this specific example. I need to do some math because I'm wondering about the movement of my water. So the first thing that I need to do is the bag is going to be 80% water. How did I figure that out? I know that it must be 100% minus 20% is 80%. Okay, so if it's 20% salt, 80% water. You just need to do some simple math. So if I have a 5% salt solution on the outside in the water, what do you think is going to be the percentage of water on the outside of this bag? If I have a 5% salt solution on the outside, what is my water percent? If you said 95% water, you are correct. Okay, 100 minus 5 equals 95. So just some simple math. So I have 80% water on the inside of the bag and 95% water on the outside of the bag. So if things move from a high concentration to a low concentration, look at my numbers here. In which direction will the water move? Will the water move into the bag, or will the water move out of the bag? Remember, from a high concentration to a low concentration, in which direction will my water move? In or out of the bag? If you said into the bag, then you were correct. So here we go from a high 95% to a low concentration. So from my 95% to my 80%, which was my lower number, so water will move into the bag. In the water solution, we actually have 3 types of water solutions. So when we're speaking about osmosis, there are going to be 3 types of solutions that your cells could be in, or these hypothetical bags. The first is going to be an isotonic solution. The second is hypertonic, and the third is hypotonic. Okay, you need to know some things about each one of these, and each one of these are examples of osmosis. So you can see here some examples. In a hypotonic solution, you can see in this little GIF here that my cell is getting bigger. Things are going into the cell, and we're going to discuss that. In my isotonic solution, you can see that there's arrows moving in and out. This guy is staying the same. He's pretty happy. And in my hypertonic solution, you can see that the arrows are leaving and my molecules are leaving and my cell is shrinking and he's not too happy. So I want you guys to kind of keep this little image, this GIF in your head. You can come back to it if you need it. It's going to be very helpful in determining the difference between these types of osmosis solutions. In isotonic solutions, the amount of water is going to be the same on the inside and the outside of the cell. Okay. The water is going to flow in the same rate in both directions in this type of solution. This is an ideal solution. Okay. Up here in all of these examples, you're going to see an animal cell and you'll also see a plant cell in the bottom. So my arrows go in and out. Again, water is moving in and out at the same rate. And this is the ideal solution for plants and animals. They both kind of want to be in this isotonic solution situation because it's helpful and it's homeostasis. Things are nice and even. We can also term this dynamic equilibrium. Okay, things are equal. Plants are happy, animals are happy, things are moving in and out as they should. In a hypertonic solution, and you're going to notice that I keep saying these kind of words when I pronounce things because it's going to help you remember. So hypertonic solutions will have more water on the inside of the cell than on the outside of the cell, okay? Now, if I have more water on the inside, that's my high concentration, right? And water is going to leave to where there's a lower concentration. So the net water of movement is out of the cell, okay? Now you can see here my animal cell is shrinking. It's getting smaller. My plant cell, the cell wall is holding it up nice and kind of square or rectangular like, but the cell membrane here will start to shrink, okay? So if you've ever seen a plant, right, and it hasn't been watered in a while and it's nice and shriveled, this is why, okay? Its cell membranes are kind of starting to cave in on itself. The cell wall is still holding it up, but it's a shriveled plant, okay? So water will move out in this situation. There is a term for this, okay? Plasmolysis is when too much water will move out and the cell will collapse. Now, this won't necessarily happen to plant cells, but it will happen to animal cells if too much water leaves. In a hypotonic solution, now I kind of use this term to remember it. I think hippo. Okay, hippos are really big. I try to remember that when I'm thinking about hypotonic solutions because in a hypotonic solutions, the cells are going to get bigger. There is more water on the outside of the cell than on the inside of the cell. So going from my high to low concentration, the net water of movement is going to be into the cell. Okay, so you can see here in my animal cell, water is moving in. In my plant cell, water is moving in. And there is a term for this when too much water moves in and the cell membrane can burst due to pressure. This is called cytolysis. Okay, remember that lysis means to burst, cyto means cell. So cell bursting, cytolysis, is when too much goes in and the cell membrane just can't handle it anymore and it bursts wide open. Doesn't necessarily happen in plant cells. Plant cells just get more alive. Okay, they perk back up. They like to be in hypotonic solutions, just not too much. Okay, again, too much water can kill a plant cell, but it won't necessarily burst as easily as an animal cell. In cells with a cell wall, so this is kind of what I was talking about, cytolysis not likely to occur, okay, because of those cell wall. The central vacuole, remember, plant cells have central vacuoles, animal cells have small vacuoles, but what will happen is this vacuole will just become extremely full of water, and this thing called turgor pressure will increase, okay? Now, all this means is that the pressure around this cell wall is going to increase. So this cell here is what we call turgid, okay? It just means that it's like very, very puffed up with water, and it will help give structure and support to a plant cell. All right, guys, what is Drake doing? We're going to find out in our next video. It looks like he is moving some molecules, maybe. I want to point out some things here. We have our extracellular space here, okay, which is going to be outside of the cell. We have our cytosol or cytoplasm, which is the inside of our cell, and he is doing something to those molecules. We're going to find out in part 2 of this series.